Assalamualaikum and greetings to our lecturer Dr. Nurul Hikmah and fellow classmates. We from Group 5 will be presenting about tumors. Tumor is a group of abnormal cells that causes lumps or growths of the body tissue. It can start growing in any part of the cells in the human body. The tumor grows and behaves differently depending on its type, whether it is malignant, benign, and precancerous. Other than that, the tumor can be identified by the immune system in the body. The emerging tumor cells can be destroyed after they are recognized as non-self or foreign antigens on the surface of the cells by the immune responses. The constant monitoring of the immune system for small tumors is known as immune surveillance. However, if the balanced cell growth and death are disturbed as well as the immune system is not functioning properly, the tumor will begin to form.
Tumor antigen is antigenic substance that produced in the tumor cell and it will trigger the immune response. Basically, there are two types of tumor antigen, which are tumor-specific transplantation antigen (TSTA) and tumor-associated transplantation antigen (TATA). TSTA are unique to tumor cell as it only present on tumor cell and it was produced due to chemical, physical, or even viral mutation. Mutation will alter the protein. Thus, the immune system will detect and eliminate the tumor cell by antigens on the surface of the cell that were presented by MHC class 1. Meanwhile, TATA are not unique to tumor cell as it can present on normal cell during fetal. However, it will express at low level on normal cell and the level will be elevated on tumor cell. The picture on the left shows that the normal cell will produce a dark red and blue genes. However, if there is any mutation or reactivation, thus the normal cell will be converted into either TSTA or TATA. In TSTA, we can see that the gene modification convert the normal gene from dark red to light red. Due to the change of gene, it transforms the normal cell to the tumor cell as there is an alterations on the protein expressions. In TATA, it can be either inappropriate expressions or overexpressions. Inappropriate expression is where there is an expression of new genes. As we can see in the picture, there's an additional black color gene. Meanwhile, overproductions of TATA due to modifications of transcription factor produce more gene compared to the normal cell.
Next, immune mechanism of tumor rejection. One of the cells involved is T cell, which is primary cell that has the responsibility for direct recognition and killing of tumor cells. It can carry out immunosurveillance, proliferate and destroy newly transformed tumor cells after recognizing the tumor-associated antigens. The T cell response to tumor is modulated by other cells of the immune system. Some cells require the presence of humoral antibodies directed against the tumor cells to initiate the interaction that lead to death of the tumor cells. Next, cytotoxic T lymphocytes. It can recognize antigens on target cells and lyse these cells. These antigens may be cell surface protein or may be intracellular protein that are expressed on the surface in combination with MHC class 1 molecules. Next is NK cells, which lack of receptor for antigen detection but can still recognize normal cells infected with viruses or tumor cells. Their tumoricidal activity is termed natural because it is not induced by a specific antigen. Class 1 MHC molecule on the surface of normal cells inhibit NK cells and prevent lysis. Thus, the decreased level of class 1 molecule expression, characteristic of many tumor cells, may allow activation of NK cells and subsequent tumor lysis. Next, NKT cells, which specialize CD1D restricted T cells that recognize lipid antigens. NKT cells lead to activation of both innate and adaptive immune cells in the tumor microenvironment. NKT cells are innate-like T cells that quickly respond to antigenic stimulation and rapidly produce large amounts of cytokines and chemokines. Next, macrophages can kill specific tumor cells when activated by a combination of factors including lymphokines and interferon. Under certain circumstances, macrophages may present tumor-associated antigens to T cells and stimulate tumor-specific immune response.
Next is dendritic cells. They play a central role in initiation of the tumor-specific immune response, and T cells will take up tumor-associated proteins, process them, and present the tumor-associated antigens to T cells in order to stimulate the CTL response against tumor. For lymphokines, they produced by immune cells that stimulate growth or induce activities of other immune cells. And for IL-12, which is produced by dendritic cells and specifically induces CTLs, it will enhancing anti-tumor immune responses. For regulatory T cells, it is normally present in the body and help prevent autoimmune reactions. They are produced during the active phase of immune responses to pathogen, and the accumulation of these cells in cancer will inhibit anti-tumor immune responses. The last one is myeloid-derived suppressor cells, which consists of immature myeloid cells and their precursors, and these cells will accumulate in large numbers in cancers and predict poor clinical outcomes in various types of cancer. Humoral immunity. In contrast to T cells cytotoxic immunity, humoral antibodies do not appear to confer significant protection against tumor growth, and most antibodies cannot recognize TAAs. Cytotoxic antibodies are directed against surface antigens of tumor cells, and these antibodies can exert anti-tumor effects through complement fixation or by serving as a flag for destruction of tumor cells by T cells.
Inhibition tumor of immune response is acts by shutting down expressing antigen on its surface of the cells. The process called as tumor immune editing. According to Han et al., tumor cell loss antigen expression through inhibition of translation activity of its mRNA, leading to higher mutative rate of tumor cells and gene instability, mutation, and deletion. Next, the downregulation of class 1 MHC expression by DNA methyltransferase enhance the tumor evasion due to the epigenetic silencing within the tumor cells. This results in the direct methylation of class 1 MHC as confirmed by Sohar-Bhatti. By that, methylation of various essential genes contributed in the suppression and downregulation of class 1 MHC expression, then allowing antigen to be unrecognized. According to Sabri and Laudel, tumor cells impair the recognition of dendritic cells to prevent NK priming by changing their expression of interleukin-6, 10, and vascular epithelial growth factor. Due to that, it leads to the lower NK cells count by decreasing the number of lymphoid progenitor cells. Other than that, tumor also inhibit T cell activation as it expresses the ligand for T cell inhibitory receptor such PD-1 that confirmed by Abbas et al. It also may secrete immunosuppressive cytokines to induce regulatory T cell resulting in immune response suppression.
I am Iman Zulaika and I will be discussing immunological approaches in cancer therapy. According to my readings, researchers have been using vaccine-based and immune cell-mediated base to cure cancer. As you can see, oncolytic virus is an example under vaccine-based therapy. However, for immune cell-mediated base, researchers have been utilized T cells and dendritic cells in targeting cancer. For immune cell-mediated based, T cells secreting interleukin-2 have been used to treat tumors in the past. IL-2 plays a critical role in the activation of immune system, which could be a useful way to eradicate cancer. As monotherapy, IL-2 has been demonstrated capable of mediating tumor regression and was approved for metastatic renal cell carcinoma and metastatic melanoma by FDA. T cells isolation have multiple approaches, which are tumor-infiltrating lymphocyte, T-cell receptor, and chimeric antigen receptor T cell. The three of them involve collecting a patient's own immune cells, then multiplying them in large numbers in the lab and infusing the cells back into the patient. Okay, what are the differences between these T cells therapy? For tumor-infiltrating lymphocyte, doctors will harvest naturally occurring T cells that have already infiltrated patients' tumors. Then they activate and expands them. Large number of these activated T cells are reinfused into patients where they can seek out and destroy tumors. Unfortunately, not all patients have T cells that have already recognized their tumors. These T cells may not be capable of being activated and expanded to sufficient numbers to enable rejection of their tumors. In T cell receptor, the T cells are equipped with a new T cell receptor that enables them to target specific cancer antigens. These cells are then multiplied, then reinfused into patients once again. TIL and TCR therapies can only target and eliminate cancer cells that are present in their antigens in a certain context. When bound to the MHC, these artificial CARs do not require MHC molecule and T cell interaction, instead of acting directly against T cells expressing tumor antigens. Dendritic cells represent a heterogeneous group of innate immune cells that infiltrate tumors and process and present tumor-derived antigen to naive T cells. Dendritic cells play a critical role in priming anti-tumor T cell immunity and thereby represent a major therapeutic target for cancer immunotherapy. Dendritic cells is stimulated ex vivo with danger signals and loaded with tumor-specific antigens on their major histocompatibility complex molecules with the intent of activating antigen-specific T cells which selectively eliminate antigen-bearing cancer cells. These are the major antigen-representing cells in the body which, once activated, present antigens to CD4 and CD8 T cells and induce protective T cell responses. If a cancer-specific antigen is presented, this can result in an anti-tumor response. As for vaccine-based, an oncolytic virus can be described as a genetically engineered or naturally existing virus that can selectively replicate in cancer cells and then kill them without damaging the healthy cells. There have been many kinds of oncolytic viruses such as herpes simplex virus, adenovirus, and Coxsackie virus.
As a conclusion, the tumor is a group of abnormal cells that grows excessively in the human body tissue. These cells are known as foreign cells to the body and needs to be eliminated before causing harm to the body. Hence, the immune responses toward the tumor cells are very important to prevent the tumor from becoming cancer and ensure the safety of the human life. These are the references used for the presentation. That's all from us. Thank you.